aerobic respiration. So okay, pyruvic acid can enter into the aerobic respiration. Okay, after the glycolysis, pyruvic acid is produced. It can enter into aerobic respiration. So this reaction occur in the glycolysis. After glycolysis, pyruvic acid is produced. That pyruvic acid can enter into aerobic respiration in most of the organisms. So from the cytoplasm, the pyruvic acid enter into the mitochondria for further oxidation. Okay, here in this case, the pyruvic acid completely oxidized in aerobic respiration. The breakdown of pyruvic acid or oxidation occur in the presence of oxygen. Prokaryotic organisms, there is no bacteria, no mitochondria. You know that in bacteria, the aerobic respiration occur in the cytoplasm and the plasma membrane, because there is no mitochondria for prokaryotes. Pyruvic acid is, I told you, is completely oxidized to carbon dioxide and water, and all the energy from the pyruvic acid, maximum energy from the pyruvic acid is released and can be converted into ATP. Okay, so that is what happening here. So here we can see from the cytoplasm, this transferred to the matrix of the mitochondria, so fluid portion of the mitochondria. Here these reactions takes place. Okay, right. So we can see what is the process here. So first it undergo a complex reaction. Okay, pyruvic acid once enter into the mitochondria, it converted into acetyl coenzyme A. So that is the first reaction. Okay, here same A. Here that reaction, carbon dioxide is released. One carbon dioxide molecule is released. Suppose if there is one pyruvic acid, one carbon dioxide released. So here we can say if 2 pyruvic acid means 2 carbon dioxide. So that is the case. So here one NADH2 molecule can also be produced from one pyruvic acid. Okay, so see here the pyruvic acid undergo decarboxylation, that means removal of carbon dioxide, and it also undergo oxidation. So that means from pyruvic acid hydrogen ions and electrons are removed, and these hydrogen ions and electrons are taken away by this NAD. Receive this hydrogen ion and electron, and it become NADH2. So that is what happening here. So here the pyruvic acid undergo oxidation. So there are two process, oxidation and decarboxylation. Okay, so that means this we can say the pyruvic acid undergo oxidative decarboxylation. That means removal of hydrogen ion and electron occur. That means oxidation and release of carbon dioxide, decarboxylation. So it so that is a complex process reaction. It occur in the presence of the enzyme pyruvate dehydrogenase complex. So there is a complex enzyme, pyruvate dehydrogenase complex. So that needs the presence of magnesium ion also. So this reaction occur in the presence of magnesium ion also required to activate the enzyme, and this magnesium Mg2 plus ion also there, and along with that along with this reaction, coenzyme A also combined, also participating in this reaction. Coenzyme A also participating in this reaction. As a result, a compound produced will be acetyl coenzyme A. So that is the product formed here. So this compound is a 2-carbon compound. Why? Because one carbon dioxide is removed from pyruvic acid. So there is remaining only 2 carbon is there. So it become acetyl coenzyme A. So that is the 2-carbon compound. Okay, produced. So this reaction usually we termed it as link reaction. Why? Because it's a reaction that occurs just before the Kreb cycle. Okay, before the substrate enter into the Kreb cycle, this reaction, link reaction. Okay, so here the product acetyl coenzyme A can also be termed it as connecting link. So it is a sub compound formed in between the glycolysis and Kreb cycle. So glycolysis end in pyruvic acid, and Kreb cycle start from oxaloacetic acid. Oxaloacetic acid is first compound of Kreb cycle or aerobic respiration. Okay, so this is the starting of point of the aerobic respiration. So acetyl coenzyme A then enter into the further oxidation. Okay, acetyl coenzyme A now enter into a series of reaction, a cyclic reaction, with termed it as Kreb cycle. It was discovered by a scientist, we know that, Hans Krebs. We can discuss it later. Hans Krebs. So we termed it as Kreb cycle. So here first step here, the first compound oxaloacetic acid is a 4-carbon compound. Oxaloacetic acid react with acetyl coenzyme A. Okay, so here in the presence of the enzyme citrate synthase enzyme, a 6-carbon compound is produced. We termed it as citric acid, the 6-carbon compound. Okay, 4 plus 2. Okay, 4 plus 2, oxaloacetic acid 4 carbon, acetyl coenzyme A 2 carbon. So a 6-carbon compound, citric acid will be produced. Citric acid again converted to isocitric acid. This compound is also there. That is not much important for you for the NEET purpose, or Harris in board exam. This is not important for you. You can directly study like this. Citric acid get converted to isocitric acid. There is no significant changes occurring here, so we can directly study like this. Citric acid we can say directly converted to the next compound known as isocitric acid. Again a 6-carbon compound with the help of the enzyme. This is the enzyme aconitase enzyme, isocitric acid. Okay, isocitric acid important, very important. Isocitric acid undergo oxidation. So is the first compound in the Kreb cycle that undergo oxidation, isocitric acid. Oxidation, we already know what is that. Here isocitric acid remove the hydrogen ion and electron. Okay, so that hydrogen ion and electron combine with what NAD and NAD. So it become NADH2. This coenzyme NAD reduced to NADH2. Okay, coenzyme reduced, but the substrate undergo oxidation. Okay, so then it become oxalosuccinic acid. The next compound is the oxalosuccinic acid, again a 6-carbon compound. Okay, oxalosuccinic acid also a 6-carbon compound. Immediately it undergo decarboxylation. So from oxalosuccinic acid a carbon dioxide is released, and it converted into alpha ketoglutaric acid. Okay, alpha ketoglutaric acid is the first 5-carbon compound. This is the only 5-carbon compound in the Kreb cycle. Very important, only 5-carbon compound in the Kreb cycle, alpha ketoglutaric acid. Then immediately then it undergo oxidative decarboxylation, similar to pyruvic acid. Here also this compound also almost the same type of reaction, similar to the pyruvic acid. Here from here a carbon dioxide molecule is released, that means decarboxylation, and hydrogen ion and electrons are removed, that means oxidation occur. Alpha ketoglutaric acid also undergo oxidation, thus NAD get converted to NADH2. So that is the oxidation reaction. So ketoglutarate acid undergo oxidation also. At the same time decarboxylation, both the process occur. Okay, removal of electron and removal of carbon dioxide, both the process occur in this step. Okay, so it is oxidative decarboxylation. This reaction also oxidative decarboxylation. So here again the same thing happen. Coenzyme A also combine with this. Okay, coenzyme, thus it become succinyl coenzyme A. Succinyl coenzyme A is the next 4 carbon compound. So note it. So this is the 4th carbon compound, but this is 5. Why? Because here carbon dioxide is removed. Okay, so some enzymes are also there, that is not that much important. The ketoglutarate dehydrogenase is the enzyme. Ketoglutarate dehydrogenase mentioned here. Succinyl coenzyme A now again undergo oxidation. Succinyl coenzyme A converted to succinic acid. Succinic acid. During that change, here you can see the coenzyme A is removed. Coenzyme A combine here. Coenzyme A is there. Okay, so that coenzyme A is removed from that molecule substrate. Okay, so it converted to succinic acid. Okay, so here during this step one important process occur. What is that? GDP get converted to GTP. So that is another thing here. Okay, that means GDP get the phosphorus from this substrate. Who is that? Succinyl coenzyme A. From here phosphorus is given to GDP, directly given to GDP, so it become GTP. Okay, right. So that is again important process. So that is substrate level phosphorylation. From the substrate, phosphorus is given to GDP, so it converted to GTP. Okay, so here we can see, so the GTP again donate that phosphorus to ADP, immediately donate that phosphorus to ADP, so it become ATP. GDP can immediately donate that phosphorus from here to ADP, so thus ATP can be produced. Alright. Okay, so succinic acid again 4 carbon compound. This is again 4 carbon compound. Succinic acid can be become fumaric acid. Succinic acid again undergo oxidation. Okay, so this is the 3rd oxidation process. Succinic acid, okay, undergo oxidation. That means here again we can see the removal of electrons and hydrogen ion. So we can see the FAD become FADH2. Okay, so here coenzyme is FAD, not the other one. Okay, so here the energy rich or here reduced coenzyme, we can see FADH2 can be produced. So another important thing is that here the enzyme for this conversion is succinate dehydrogenase complex. Succinate dehydrogenase. So why? Because this enzyme, this is important. This enzyme is attached to the inner membrane of the mitochondria. The remaining all enzymes of this Kreb cycle, all enzymes are present in the fluid part or matrix of the mitochondria, except this one. So this one is present only in the on the inner membrane of the mitochondria. Okay, who is that? Succinate dehydrogenase. Okay, so thus the next compound fumaric acid is produced. Okay, right, fumaric acid. So then remaining 2 more steps are there. Fumaric acid get converted to malic acid. Same, not important. Fumarase, malic acid in the last step, malic acid become oxaloacetic acid. Okay, malic acid get converted to oxaloacetic acid. Again this is a oxidation process. Malic acid undergo oxidation again and produce oxaloacetic acid. Here again coenzyme NAD receive that electrons and it get converted to NADH2. Coenzyme reduced, NAD reduced to NADH2. Okay, right. So here the enzyme malic dehydrogenase. Okay, right. So NAD become NADH2. Okay, so this is the overall process, so that cycle continue like this. So again oxaloacetic acid is regenerated, then again it can combine with acetyl coenzyme A, and the cycle can continue like this. The same process repeat. So this is the cyclic set of reaction. It was discovered by Hans Krebs. You know that Hans Krebs. So this cycle reaction, anaerobic respiration, can also be termed citric acid cycle. So why? Because the first compound is a citric acid, produces citric acid cycle or tricarboxylic acid cycle, right? Like that. So this is the main process of aerobic respiration. So during this process, so many important points are there you need to remember. So how many oxidation pyruvic acid undergo, how many oxidation? So you can see pyruvic acid undergo how many oxidation. So here one first oxidation here, first oxidation during the conversion or first conversion into acetyl coenzyme, there an oxidation occur. So here then pyruvic acid under the second oxidation, second oxidation here, third oxidation here, fourth oxidation here, and this is the fifth oxidation. So totally we can say the pyruvic acid once enter into the mitochondria undergo, there are 5 oxidation, it passes through 5 oxidation steps. Okay, during the oxidation breakdown, 5 oxidation steps are there, and there are 3 decarboxylation process. Pyruvic acid, you know that having 3 carbon atoms are there. All the 3 carbon atoms are removed in successive step. The 3 carbon atoms are removed in the form of carbon dioxide. So here one carbon we can see removed as carbon dioxide in the first step. So then next carbon released here from here, and next carbon here from here oxalosuccinic acid, and here one more carbon, 3 carbon atoms are removed like this. Okay, so pyruvic acid we can say undergo 5 oxidation and 3 decarboxylation. Pyruvic acid, when we consider pyruvic acid. Okay, but the Krebs cycle start from here, from oxaloacetic acid onwards. So when we consider this cyclic reaction, how many oxidation and decarboxylation we can say? In short form we can say IKSM. So you can see here IKSM, which all molecules undergo oxidation, how many oxidations are there, which all molecule undergo oxidation. Isocitric acid undergo oxidation, you can see here isocitric acid undergo oxidation. Okay, ketoglutaric acid undergo oxidation here. Okay, then IK ketoglutaric acid, alpha ketoglutaric acid undergo oxidation, then succinic acid undergo oxidation again, then malic acid undergo oxidation. So there are 4 oxidation steps. So that what are the steps? Which all molecule undergo oxidation? Isocitric acid, IKSM. You can in short form you can remember like this. Then ketoglutaric acid, succinic acid, and malic acid. So these are the 4 molecules, intermediates that undergo oxidation. Okay, 4 oxidation reactions we studied here in this cyclic reaction, and oxidation 4, and how many decarboxylation? 2 molecules undergo decarboxylation, 2 decarboxylation. Oxalosuccinic acid you can see here, and ketoglutaric acid. So 2. Okay, so that is the answer. Okay, that is oxalosuccinic acid and ketoglutaric acid. Both the molecules undergo decarboxylation. So that is an overall process here, Krebs cycle 4 oxidation and 2 decarboxylation. So what is the net product? So how many okay, we can see what is the energy balance of this Krebs cycle, or okay. So in Krebs cycle we can see how many NADH2 molecules are produced. Here one NADH2 from, suppose if it is so we can start from pyruvic acid. From pyruvic acid, how many NADH2 molecules can be produced? One pyruvic acid. You consider one pyruvic acid. Here one NADH2 in this step, then second NADH2, third NADH2, and fourth NADH2. So from one pyruvic acid, 4 NADH2 molecules are produced. One pyruvic acid, 4 NADH2 molecules are produced. Then how many FADH2? One FADH2 from succinic acid. Okay, so one FADH2 is produced, and one GTP. GTP is also, we already studied, one GTP. So the total this much energy can be energy rich compounds can be produced from one pyruvic acid. Okay, so when we convert into, suppose if we are converting it into energy ATP, we can it will appear like this. One NADH2 means equivalent to 3 ATP. So 4 into 3, 12 ATP can be generated. Okay, FADH2 when it consider it can produce only 2 ATP. One FADH2 carry an energy equal to 2 ATP. So 1 into 2, 2 ATP. So but 1 GTP equal to 1 ATP. So that is 1. Okay, so totally 15 ATP can be generated, and energy equal to 15 ATP can be released from one pyruvic acid. From one pyruvic acid. If it is 2 pyruvic acid, 30. You know that one glucose molecule means 2 pyruvic acid, right? Okay, so 2 pyruvic acid means 30 ATP. 2 pyruvic acid means 2 pyruvic acid means 30 ATP. Okay, so you know that from glycolysis we can the cell can derive 8 ATP. So the totally 38 ATP can be derived from one glucose molecule. So that is the total energy balance, 38 ATP. Okay, 38 ATP, right? So if you are considering the Krebs cycle only, you need to start from this here on only, from oxaloacetic acid only. You consider, you calculate the energy balance like this. There occur 3 oxidation and 4 oxidation and 2 decarboxylation. 4 oxidation, 3 NADH2 and 1 FADH2. From one pyruvic acid means in Krebs cycle there are 3 NADH2 and 1 FADH2. Okay, based on the question we can answer the answer it. Okay, so that is the overall process of Krebs cycle, right?